Today we are going to see a topic, immunology. So immunology, it means it's a branch of biology that is concerned with immunity. It deals with the study of process by which the body defends itself from the invasion and attack of foreign organisms. See the history of immunology, Thucydides 430 backward for he invented a medicine for plague, then variolation in 15th century, then Edward Jenner, he introduced the vaccination for smallpox in the year of 1749 to 1823, whereas Louis Pasteur, he is called the father of immunology. He invented many vaccines such as pebrine, anthrax, chicken cholera and rabies. And finally, Alexander Fleming in 1881 to 1995, he also introduced some vaccines towards the immunity. So immunity, that is resistance to infection, this process is carried out by the process of recognition and disposal of the antigens, so that is foreign material that enter into our body. See, there are types of immunity. Majorly, broadly, the immunity is classified into two types, that is innate immunity and acquired immunity. Whereas acquired immunity is further classified into two types, which is called active and passive, whereas active is further classified into natural active and artificial active, and passive is further classified into natural passive and artificial passive. Next, innate immunity. What is innate immunity? See, all the living organisms are gifted with resistance to some certain infection by birth itself. So it occurs from naturally. So it is called natural immunity or innate immunity or non-specific immunity. See, there are some factors that induce the innate immunity. First, we see about physical and mechanical factors of innate immunity. Some of the physical and factors are skin. Skin is the largest organ in our body. It protects from invading the pathogens entering into our body. So it is one of the major innate immunity. Then mucous membrane, it is present, it is a collateral-like substance, a thin wall membrane present in the all opening areas. So this mucous membrane is responsible for trapping the microorganisms that enter into our body. Then cilia. The cilia is here, the small projections. It is present inside the intestine of our human stomach, which also trap the microorganisms that enter through the food into the intestine. Then coughing and sneezing. It is a mechanical factor. Through the coughing and sneezing, we can expel. When the microorganisms excessively enter into our expander in our lungs, which is expelled by the mechanical factors such as coughing and sneezing. Then peristalsis. It is a movement occur in the large intestine area to wash away the microorganisms or dust particles through the stool formations. Then washing actions of tears, saliva and urine. These mechanical factors will wash away the excess of microorganisms and also some foreign materials that enter in our body. Then biochemical factors. Then what are all the biochemical factors that responsible for inducing the innate immunity are secretion of skin. See the sweat under the pH, that is a basic pH, it is protect our skin from invading of microorganism and dust particles. Then secretion of digestive tract. Then human body, then human stomach, which secrete the highly concentrated acid called HCl, which is responsible for killing the microorganism and also for digestion of food materials. Then human milk. The human milk contains a substance, chemical substance called lactoferrin and neuroaminic acid. It gives protection to the baby for the microorganism, that is to protect the infections. Then lysozyme. It is an enzyme which is present in the tears, saliva and human milk. It is responsible for killing the microorganisms. Then interferons. See IFN, it is called IFN. It is a group of non-toxic glycoprotein produced by all the cells in body in smaller amount. This is an excellent antiviral agent. It will be in inactive form during the normal condition. Whenever our blood is infected with the virus, it will get activated, it kills the viruses. Then INFS also has a NK cell activity, that is it offering the immunity. It offering the immunity to cancer by blocking the growth of cancer cells. So we can commercially prepare interferons using the E. coli genome by DNA recombinant technology. So the IFN is grouped into IFN alpha and beta. They are produced by our T cells. Then complement. It is a group of cascade proteins found in the blood. It is responsible for the action of antigen antibody reactions. Then complements are operated by two major mechanisms, that is namely classical pathway and alternative pathway. The main function of this complement is promoting phagocytosis. Phagocytosis is nothing but a cell eating process, that our body cell eat the foreign material and it engulf it. Then properdin, it is a group of proteins present in the normal serum and it is involved in the resistance to infections. Along with the properdin, magnesium also causes lysis of gram-negative bacteria and also some viruses too. Then secretion of flora, that is our intestine. Normally our intestine consists of some floras, that is some worms. that worms will secrete some chemical substance which is called bacteriocins and acids, which also protect the invade of other microorganisms in our stomach. Then semen. Then the semen is a chemical substance produced by the male reproductive system. It secretes a chemical substance called sperma and enzyme from it for its protection. Then acute phase proteins. These are some non-toxic soluble proteins present in our blood. It is also responsible for killing the invading microorganisms in the blood. Next please. Then we go to the cellular factors. There are two cellular factors responsible for giving the immunity power to our body. See, first is phagocytosis. The phagocytosis, I already explained that phagocytosis is a cell eating process. It is done by the phagocytic cells. So, it was discovered by the scientist called Metchnikoff in 1838. There are two macrophages cells are having in our body, that is microphages and macrophages. See, macrophages are called as polymorphonuclear leukocytes such as neutrophils, eosinophils and basophils. These are the dominant white blood cells present in our bloodstream. So, these are multilobed nuclei. It is not divided, but it is very short-lived. It can be occurred only for 2 to 3 days in our blood. Then macrophages. So, macrophages is a large cells. So, it is a mononuclear cells derived from our bone marrow and differentiated into monocytes of the blood. They are long-lived and are eventually engulfed microbes. They can be withstand for 120 days. Next, the macrophages further classified into two types, that is free macrophage and fixed macrophage. See, for example, for free macrophages is alveolar, peritoneal in lymph and other tissues, whereas fixed macrophages, Kupffer cells in liver, osteoblasts is in bone marrow and also some lymphoidal tissues. Next, this is the diagrammatic representation which represents the stepwise phagocytosis action. See, in the first step, chemotaxis. That orange color cell is our cell. Then that yellow color cell is the infected, that is microorganism entering in our body. At first step, the macrophagic cell will recognize and move closer towards the microorganism. That is called chemotaxis. Then in the second step, in the second diagram, the number 2, here adherence through the PAMP recognition. Through some PAMP is nothing but a surface receptor. To the surface receptor, our phagocytic cell will go and adhere, slightly attach its surface towards it. So, that is the second step. Next, the third step is membrane activation through the danger signal. Then, immediately after the adherence of the microorganism, our phagocytic cell will activate and it forms here some structural changes in its membrane. Then, the fourth step is initiation of phagocytosis. Then immediately our phagocytic cells will form a false pseudopodia.